Welcome back to part 2 of structure and function. Now in this part we're going to explore how substrates can bind to enzymes. So in this section we're going to briefly review the function of enzymes and then discuss how a molecule can bind to its target. Now this will lay the foundation for future sections which discuss the different ways in which enzyme activity can be inhibited or modulated and how we can exploit enzymes as drug targets. Now the recommended section for the whole of the structure and function lecture series, so the lectures that took place in week 9 and in week 11, is the Introduction to Medicinal Chemistry by Patrick, and this is available in the library. So first of all, what are enzymes? Enzymes are proteins that catalyze chemical reactions within the body. And they do this by lowering the activation energy of a particular chemical reaction. Now this means they can either speed up or increase the rate of a chemical reaction that already happens, or they can initiate and control new chemical reactions. And they really are the workhorse molecule within the body. They have a role in every chemical reaction which happens within a biological system. So what happens when a substrate binds to an enzyme? Well, here we have the substrate which is entering into the active site within an enzyme. When it does this, the substrate will bind to the enzyme and interact with chemical groups present within the active site. The next step is that the reaction occurs. So this is where the substrate undergoes a chemical reaction within the enzyme active site. Now this could be something like the cleavage of a chemical bond, or it could be the formation of a new bond. Now in this case we have the cleavage of a chemical bond that allows the first product to move away from the enzyme after the reaction has completed. But this leaves a second component of the molecule within the active site. And so the final step is release. This is where the enzyme will release the product from the active site back into the biological system. Now at this point, the enzyme is regenerated and it can therefore undergo the reaction again. So if another substrate molecule enters into the active site, the enzyme can then perform the same reaction. Now the way in which a chemical reaction happens, and this is just a diagram of a chemical reaction, is that you have a starting material, then you have an energy barrier that must be overcome in order to be able to produce your final product. Now this energy barrier, which can be visualized here, is called the activation energy. And it's the energy that the system must be provided with to enable that chemical reaction to occur. Now, the way an enzyme works is it provides an alternative route for that reaction. The new route for the reaction will require less energy for that reaction to occur than it would need without the enzyme. So the way that the enzyme works is that it provides an alternative reaction route for a particular chemical reaction. And it provides a route that requires a lower activation energy, or a lower amount of energy to carry out. Now enzymes also provide a reaction surface and environment for the reaction. So they provide a binding site in which the reaction can occur. And the enzyme binding site will position the reactants in the right position, in the best position for the reaction to occur. So it will use its own chemical groups to reorient the substrate and place it in the correct position for the reaction to occur. Now it can do this by weakening chemical bonds within the substrate, or it could react to form new chemical bonds within the substrate. But ultimately it will do this by weakening other bonds. Now the enzyme may participate in the reaction mechanism itself, so it may be that functional groups within the enzyme directly react with the substrate, or they may just provide a scaffold for the reaction to occur. Now, the active site of an enzyme must be near the surface of the protein molecule to allow the substrate to enter into it. So it must be near the surface to allow access. Now, enzyme active sites are often hydrophobic. Now, the reason for this is that they facilitate reactions that are unlikely to occur in an aqueous environment, but also because water molecules can get in the way of a substrate and prevent it from binding if these water molecules are bound within the active site. And so having a hydrophobic active site means that the water molecules will not get in the way of the substrate binding into that site, and so they often tend to be hydrophobic. Now, amino acids in the active site are very important. Certain amino acids and sequences of amino acids are very good at catalyzing certain reactions. And this means that what you see as you look between the enzymes in different species is that the sequence of the amino acids within the active site is often very similar or even the same. And the reason for this is because amino acids all have individual chemical properties that make them good at catalyzing specific reactions. Now, there are two roles for amino acids within the active site. We have amino acids that are able to bind and hold the substrate within the active site, and we have amino acids that actually take part in the chemical reaction, and so they have a catalytic role. Now, here we can see an example of an active site and a selection of amino acid side chains that are present within that site. And so, for example, groups such as carboxylic acid groups or amino groups may form hydrogen bonds with the molecule to hold it in place. This could then allow other side chains such as histidine or other carboxylic acid groups to then catalyze and take part in the reaction. But each amino acid will have its own distinct role. It will either take part in the binding process, so it will hold the molecule, the substrate, in the active site, or it could take part in catalyzing the reaction directly. And there is, of course, nothing to stop an amino acid acting as both, first helping a substrate to bind, and then helping to catalyze the reaction. If we know what the substrate looks like, we can begin to have an educated guess as to what the functional groups within that substrate do when they go into the active site of the enzyme. So we can have a guess as to the interactions that are formed between the substrate and the enzyme. So let's take an example here. This is pyruvic acid, which, when it's in the presence of a reducing agent, can be processed by lactate dehydrogenase to form lactic acid. Now, this involves the conversion of this carbonyl group into an OH group, and also adds another hydrogen onto this carbon center. You should also note that it turns this carbon center here into from one that isn't chiral into one that is. Now, if we look at the functional groups present on the pyruvic acid, what we can see is that we have a carbonyl group, an acid group, carboxylic acid group, and a methyl group here. And we can have a guess at the possible interactions these groups could form based on our knowledge of how these groups can interact with other functional groups. So, for example, we know that carbonyl groups can act as hydrogen bond acceptors because the oxygen has lone pairs that are available to interact with a hydrogen bond donor. We know that methyl groups can form van der Waals interactions with other aliphatic groups, so other groups like methyl groups, CH2 groups, etc. And we know that carboxylic acid groups can lose their proton. So when the proton is released, this leaves a negative charge within the group. Now, wherever we have a charge on a functional group, if we bring this close to a functional group with the opposite charge, so in this case, if we brought this negatively charged group near to a group that has a positive charge, we can form an electrostatic interaction or an ionic interaction with that group. So in this case, if we brought this negatively charged group close to a positively charged group, we could form an interaction. Now indeed, from looking at these three groups, this is what actually happens in the active site of the enzyme. So we have a hydrogen bond formed from a donor group, so we have our OH group here, and this forms a hydrogen bond with the carbonyl, which acts as an acceptor. We also have our van der Waals interactions here with the methyl group on the molecule, and this helps hold the substrate in place. Now we also have an ionic bond that can form between a positively charged amine side chain and the carboxylic acid group as well. And so simply by knowing the functional groups and what interactions they can have with other groups, we can have an educated guess as to the types of interactions that we could see within the active site. And we can extend this to much more complicated molecules as well. So for example, here is a molecule of imatinib. Now this molecule has an array of functional groups present. It has heteroatoms incorporated into six-membered rings. It has NH groups, it has a carbonyl group here, and a whole host of other chemical features. But by knowing what interactions these groups could have, we can have a guess as to what happens in the active site. So for example, this NH group here could act as a hydrogen bond donor, just as this group can. This nitrogen here will have a lone pair on the nitrogen, and it will be able to act as a hydrogen bond acceptor. Now we also have regions with six-membered rings that contain predominantly CH groups, and these can be hydrophobic, so they could interact with hydrophobic regions within the active site. Now if we have a look at what actually happens in the active site, a lot of what happens mirrors our expectations based on the functional groups that are present within the molecule. So this is what the active site looks like, and we can see here that the nitrogen will form a hydrogen bond with an NH group from the peptide backbone within the enzyme active site. So this NH group here acts as the hydrogen bond donor and this group as the acceptor. We can see that these two rings will interact within a hydrophobic pocket, and so they form a hydrophobic interaction with the active site. We also have the NH here, which we thought could act as a hydrogen bond donor, and indeed it does. It acts as a hydrogen bond donor with an OH group from threonine. Now remember, OH groups can act both as a donor and an acceptor. We also form other interactions as well, such as another hydrogen bond here, and here as well, of course, where this group is acting as a hydrogen bond donor, and this carbonyl is acting as an acceptor. And we have another hydrophobic pocket that can accommodate this group as well. And so you're not expected to be able to tell what all the binding interactions will be simply by looking at a molecule. However, you should start to think about what the properties of each functional group are, and maybe some of the possibilities for interactions that it could have. You'll never be able to predict all of the interactions that can occur, but by looking at the substrate molecule, we can start to get a feel for perhaps what sort of interactions will be present within the active site. Now this takes us on to the theory of induced fit versus the theory of lock and key. Now both of these hypotheses describe how a substrate can bind to an enzyme. Now the first hypothesis is the lock and key hypothesis, and you may have encountered this before, perhaps at A level. Now this theory states that the substrate binds into the active site of the enzyme, and that the substrate is a perfect fit to bind into that active site in the same way that a key is a perfect fit for a lock. Now it says that the substrate and the enzyme will have a very rigid structure and that they will simply be a perfect fit. Now the problem with this theory is that it breaks down when we realize that enzymes are able to turn over multiple substrates. So it might be that one enzyme molecule is designed to react with a particular substrate. However, in reality, there is some flexibility around this. So actually, substrates or molecules that are very similar to the substrate could also be processed by the enzyme. So if we're not talking about large changes to the molecule, usually enzymes are able to still react and turn them over to a certain extent. It may be that the reaction takes longer, it happens with a lower rate, but enzymes can turn over, they can react with multiple substrates. Now, if they could only react with the molecule that is a perfect fit into their active site, this lock and key hypothesis would be considered. But because they can turn over multiple substrates, the lock and key hypothesis breaks down. Now instead, there is an alternative hypothesis, which is Kostrom's theory of induced fit. Now what this says is that when the substrate binds into the active site of the enzyme, that the substrate is not an ideal shape to fit into the active site. And so the enzyme is forced to change and forced to wrap around the substrate in order to facilitate the reaction. So the active site is forced to change, and it's forced to change by the maximization of bonding interactions between the substrate and the enzyme. Now this explains why an enzyme is able to react with multiple substrates even when these are technically different, even when they're chemically different. So it explains why a reaction can occur between a substrate and an enzyme and between multiple versions of the substrate. And so it's Kostrom's theory of induced fit that is of most relevance when we're looking at binding interactions between molecules and their target, be it substrates binding into enzymes or messenger molecules binding to receptors, which we'll look at later on in this lecture series. So what happens during the process of induced fit? Well, here we have an example, the same one that we had before, showing our molecule entering into the binding site of an enzyme. Now within this binding site, the molecule is able to have a number of different interactions. So the methyl group can form van der Waals interactions with groups within the amino acid side chains present in the binding site. The carbonyl group here can act as a hydrogen bond acceptor, accepting the hydrogen from an OH group over here, which acts as the donor. And our carboxylic acid group contains a negative charge, which can interact with the positive charge on a positively charged amine group here. Now when these groups interact, they draw the groups in the enzyme closer to them. So we can see here that the region having a van der Waals interaction has caused the active site to wrap around this methyl group. We can see that this hydrogen bond here has shortened, so it's gone from this distance to this distance here, and that's pulled the protein over and around the molecule. And the electrostatic interaction or ionic bond that occurs here has also drawn the amine group closer to the molecule. So the change in shape in the protein is driven by maximizing the intermolecular binding interactions. So these are the binding interactions between the molecule and the groups in the active site. Now this process explains how a range of substrates can be tolerated by the enzyme and how the enzyme can turn over or react with a range of substrate molecules. Now you should note that the substrate is not a passive spectator in this process. It doesn't just simply watch whilst the protein wraps around it. The process is driven by interactions between the molecule and the protein. So the role of the substrate molecule in this isn't passive. It plays an active role in changing the shape of the protein. Now what we can have is bond rotation that can occur to fix the substrate in different conformations. So for example, this substrate molecule could freely rotate around this carbon-carbon bond here. And if the correct binding interactions here and here were not in those places, this molecule could freely rotate. And the same goes for any single bonds within the molecule. You can get rotations around those bonds, which could end up with the groups in different positions. Now, sometimes this will place the molecule in the best position, and other times it won't. Now the substrate has to be held in position for the reaction to occur. So that is why, for example, this relatively small molecule has 3 different binding interactions with the active site. Now fixing this molecule in position sometimes helps to weaken the bonds that are involved in the reaction. So in this molecule, this carbonyl group here is converted into an OH group and a hydrogen coming off this carbon. The formation of a hydrogen bond between the carbonyl group and this hydrogen here actually weakens this oxygen-carbon double bond, and that is what allows the reaction to take place. Now the binding interactions that the molecule has with the active site need to be strong enough to hold the substrate in, but they also need to be able to be broken to allow the release of products. So what we do not want within an enzyme is a substrate that binds permanently. Now in the next section we'll briefly discuss how enzymes can be used as drug targets.